Base pair beef, a nun's finger bone, and bunions are usually left out of the story of the identification of the remains of the Romanovs, members of the dynasty that ruled Imperial Russia for over 300 years. It's a story that sits on a foundation of mitochondrial DNA analysis, not without some controversy, but it's actually much more complicated than that. The identification of the Romanovs was a major moment in the study of ancient DNA and how we can use it to answer questions about the past. But it's also a story that is still shaping the fields of molecular anthropology and forensic science. This video series will be the deepest dive into the frankly bonkers, but undeniably fascinating story of Romanov DNA that nobody asked for. The Russian Revolution and the fall of the Romanov dynasty is a story that has been told so many times. And I'm not going to really spend a lot of time on the historical background here. There are more in-depth videos and books about the end of the Romanov dynasty. We're gonna speedrun this so we can get to the DNA stuff. The reign of Nicholas II, the last Tsar of Russia, was a disaster. On top of all of the problems Russia already had long before he came to the throne. With the First World War tearing Europe apart and Russia being a total mess, Nicholas abdicated in 1917. He also gave up the throne on behalf of his son, the Tsarevich Alexei. The throne was then offered to Nicholas's brother Michael, but he absolutely did not want that, and he refused, ending Romanov rule of Russia. Nicholas and his immediate family, which consisted of his wife Alexandra and their 5 children, Olga, Tatiana, Maria, Anastasia, and Alexei, were placed under house arrest. Eventually they were taken to the city of Yekatarinberg, where they lived in the Ipatiev house with the 4 servants who had remained with them. The family physician Dr. Eugene Botkin, the valet Alexei Trupp, the maid Anna Demidova, and their cook, Ivan Kharitonov. Early in the morning of July 17, 1918, Nicholas, Alexandra, their 5 children, and the 4 servants were executed by firing squad in the basement of the Ipatiev house. A grave was discovered in the forest approximately 20 miles from Yekaterinburg in 1979 by Julie Ryabov and Alexander Avdonin. However, the idea that this grave could be that of the Romanovs meant that the Soviet Union had to fall before things calmed down in terms of Russia's political situation, and then an excavation took place. That excavation took place in 1991. Archaeologists excavated a shallow grave that contained the remains of 9 adult individuals. This is a bit of a problem, because 11 people died in the basement of the Ipatiev house on that July night in 1918. Nicholas, Alexandra, their 5 children, and 4 servants. What did the skeletal remains tell us about the grave's 9 occupants? The gold, platinum, and porcelain dental work some of them had suggested that these remains belonged to aristocrats. Those sound like some expensive metals and materials to have in your mouth. The skeletons showed a lot of evidence of trauma, including injuries consistent with bullet and bayonet wounds. This trauma does fit with the brutal descriptions of the execution of the Romanovs and their staff. There was also evidence of these individuals' faces having been mutilated and destroyed, possibly in an attempt to prevent their bodies from being identified. Alright, what about the DNA? Buckle up. This is going to be a lot, and we're going to revisit these methods and the results of this first study multiple times. DNA testing in archaeological context was still a very new thing in the early 1990s. In terms of DNA testing in general, the Human Genome Project, the great big quest to map and sequence the human genome using nice, shiny samples from modern living humans, had only been launched in 1990, a year before the excavation of this grave. Even with this study taking place during the early days of ancient DNA, this study accomplished a lot. So what did various ways of analyzing DNA tell us about these individuals? Let's look at a specific gene first, the amelogenin gene. Amelogenin is a protein that has an important role in the development of tooth enamel, and the amelogenin gene codes for that protein. Mutations in this gene causes X-linked amelogenesis imperfecta, which results in abnormal or even absent enamel, which in turn will lead to other issues with the teeth. Are any of these fun facts relevant to the individuals in this grave? No. It's where this gene lives within the human genome that makes this gene important when it comes to collecting data from unidentified human remains. When we inherit genes, we inherit 2 copies of each gene. You get 1 copy from your biological mother and the other copy from your biological father. These genes live on chromosomes, and you receive 23 chromosomes from each parent for a total of 46. One pair of these chromosomes is referred to as sex chromosomes, because they play a role in the development of the characteristics we associate with being biologically female or biologically male. Most of the population will have either one X and one Y chromosome, male, or two X chromosomes, female. This is because you get your first X chromosome from your mom and your second sex chromosome from your dad, who can give you either X or Y. This is just the basics. It can get a lot more complicated than that, but your copies of the amelogenin gene live on the X and Y chromosomes. The different copies of the amelogenin gene, the X copy and the Y copy, are different sizes. When we talk about the size of a gene, we mean how long is the string of base pairs that creates that gene. The X version of the amelogenin gene is 106 base pairs long, and the Y version is 112 base pairs long. Those bases that make those base pairs are the nucleotides adenine, thymine, guanine, and cytosine, and usually I explain gene construction the other way around. I have a video that explains the basics of DNA in a way that isn't backwards, but the different sizes of the X and Y versions of amelogenin makes this gene useful. We're trying to figure out if a skeleton is that of a biologically male or biologically female individual. Alright, let's get back to those individuals in the grave near Yekatarinberg. Looking at the amelogenin gene of those 9 individuals gave a result of 4 male individuals and 5 female individuals. If we're comparing this to what we would expect if these are the Romanovs, we're missing 1 male and 1 female. If they are the Romanovs, we should expect some of these individuals to be related to each other. How can we use DNA to find those genetic relationships? Short tandem repeats, or STRs, are little chunks of DNA between 1 and 6 base pairs long, and they're repeated multiple times in a person's DNA. There are specific locations for STRs within the human genome, and the number of repeats of these little chunks of DNA varies from person to person. Your particular profile of STRs is inherited from your parents, so you get some of your STRs from mom and some from your dad. Short tandem repeat analysis of these 9 individuals did find a family group, with the skeletons labeled 3, 5, and 6, all female individuals, sharing patterns with skeleton 7, another female individual, and skeleton 4, a male individual, that we would expect to see if skeletons 4 and 7 were the parents of skeletons 3, 5, and 6. So here's what we are looking at with that family group identified in that grave. We'll start with skeleton 7. That is a female individual. We have skeleton 4. And they're a male individual. And so they're sharing these short tandem repeat patterns with 3 other skeletons, 3 other female skeletons in this group. We have skeleton 3. Skeleton 5. And skeleton 6. And again, these 3 individuals are all female. So we can also do mitochondrial DNA and compare these 3 individuals with skeleton 7. If skeleton 7 is their biological mother, they're going to share the same mitochondrial DNA sequences as well as half of their STR patterns. The other 4 adult skeletons, 1, 2, 8, and 9, were excluded as possible parents of skeletons 3, 5, and 6 because they didn't share the right patterns of short tandem repeats. So if these are the Romanovs, we would have Tsar Nicholas II, skeleton 4, his wife Tsarina Alexandra, skeleton 7, 3 of their 4 daughters, 3, 5, and 6, and the family's 4 servants, genetically unrelated to them. We're still missing one of the daughters and Nicholas and Alexandra's son, Tsesarevich Alexei. To further explore kinship and genetic relationships between those individuals and possibly store their names to them, geneticists tested and analyzed their mitochondrial DNA sequences. Mitochondrial DNA is unique and really useful for a number of reasons. All of your DNA lives inside the cells of your body, with the exception of red blood cells, which don't have a nucleus, and egg and sperm cells, which only carry half of your DNA. In the rest of your cells, humans have 2 genomes, the nuclear genome that lives in a cell's nucleus, and the mitochondrial genome, which lives in the mitochondria, the powerhouse of the cell. Your nuclear DNA, also called autosomal DNA, is all of the genetic material you inherited from your biological parents. Mitochondrial DNA, however, is only inherited from your biological mother. That makes it useful for tracing maternal lineages. 6 different mitochondrial DNA sequences existed across these 9 individuals. The sequence of skeleton 7 matched the mitochondrial DNA sequences of skeletons 3, 5, and 6. This is what we would expect if skeleton 7 belonged to Tsarina Alexandra and the other 3 skeletons belonged to her daughters. But this still isn't enough to identify them. To do that with mitochondrial DNA, we need genetic material from someone maternally related to Tsarina Alexandra. Alexandra was born Princess Alex of Hesse-Darmstadt in Germany. Her parents were Grand Duke Ludwig IV of Hesse-Darmstadt and Princess Alice of Great Britain, herself a daughter of Queen Victoria. The mitochondrial DNA lineage shared by Victoria, Alice, Alexandra, and other female descendants of Queen Victoria is sometimes referred to as the Victoria or Queen Victoria lineage. The nice thing about doing these kinds of studies with royalty is the genealogical records that help us find people to whom we can compare DNA samples, because that's what we need when we're trying to use DNA to restore a name to an unidentified individual. Alexandra's mother, Princess Alice, had multiple daughters. One of those daughters, another Victoria, had a daughter, another Alice, who became known as Princess Alice of Greece and Denmark after her marriage. This Alice's son, Prince Philip, Duke of Edinburgh, husband of Britain's Queen Elizabeth II, donated a DNA sample for this study because he would share that same mitochondrial lineage descended from Queen Victoria. Welcome to the Queen Victoria mitochondrial DNA lineage, in which half the people are named either Alice or Victoria. Let's start with Queen Victoria. She had a bunch of daughters, one of whom was Princess Alice. Alice married Ludwig, Grand Duke of Hesse, and they also had multiple daughters. They named their oldest daughter Victoria because that's actually what a lot of Victoria's kids did, to be honest. And then she had a daughter, also Alice. But then Alice had a son, Philip, Prince of Greece and Denmark. He actually had to get rid of his Greek citizenship in order to then become the Duke of Edinburgh and marry the future Queen Elizabeth II of Great Britain. But then we also, of course, have Alexandra. And then she and Nicholas had their 5 children. Olga, Tatiana, Maria, Anastasia, and Alexei. So, all of these people descended from Queen Victoria are going to have the same mitochondrial DNA sequences, the same mitochondrial DNA lineage. His mitochondrial DNA sequence matched the sequences of skeleton 7, the putative Tsarina Alexandra, and skeletons 3, 5, and 6. What about skeleton 4, the putative Tsar Nicholas? We already know that this is a male individual who shares short tandem repeats with skeletons 3, 5, and 6, suggesting a parent-child relationship with them. However, their mitochondrial DNA sequences aren't a useful comparison if skeleton 4 belongs to Tsar Nicholas. We need people who would share his sequences to compare to skeleton 4. So, the geneticists found 2 living relatives who can trace their unbroken maternal lineages back to Nicholas's grandmother, Louise of Hesse-Kassel, Queen of Denmark. This article doesn't provide the names of these living relatives. They might have wanted to remain anonymous, but they are a great-great-grandson and a great-great-great-granddaughter of Queen Louise. The mitochondrial DNA sequences obtained from those 2 living relatives of Tsar Nicholas match the sequences from skeleton 4. Except for one thing. A mutation called heteroplasmy. Heteroplasmy is the presence of more than one type of mitochondrial DNA sequence, called a mitochondrial DNA haplotype, in a cell or an individual. This isn't the same thing as contamination, where you have multiple mitochondrial DNA sequences originating from different people that got into the sample when they're not supposed to be there. Heteroplasmy showed up in multiple DNA extractions from skeleton 4, so the geneticists made sure to replicate that and verify that this mutation really was present in this individual's DNA. So, what does this mutation look like? To understand the heteroplasmy observed in skeleton 4, we have to revisit nucleotides and base pairs. The 4 nucleotides, adenine, thymine, guanine, and cytosine, or A, T, G, and C, are the most basic building blocks of DNA. They're the nucleic acids referenced in the name, deoxyribonucleic acid, and we also call them bases. These bases pair up with each other, giving us base pairs for double-stranded DNA. A pairs with T, and C pairs up with G. Line up a given number of these base pairs together in a specific order, a specific sequence, and you've got a gene. It is more complicated than that, but that's a discussion for another video. The human mitochondrial genome has 16,569 base pairs. Although the paper that told me that is already 10 years old, stuff gets outdated so fast. But the heteroplasmy observed in skeletons for mitochondrial DNA sequences is at one of these nucleotide positions, the place where a nucleotide sits in the DNA sequence. At position 16169, skeleton 4 had 2 nucleotides, cytosine and thymine, instead of only 1 nucleotide. At the time of this study, which was published in 1994, heteroplasmy was thought to be rare, and it's the only difference between the mitochondrial DNA sequences of skeleton 4 and Tsar Nicholas's living relatives. The DNA of Nicholas's mother, Empress Maria Fyodorovna of Russia, born Princess Dagmar of Denmark, and his maternal grandmother, Queen Louise, wasn't available at the time of this study to see if they also had heteroplasmy at this nucleotide position, and then maybe that mutation just wasn't passed down to the living relatives. Working with the idea that Tsar Nicholas had heteroplasmy, putting all of this genetic data together led the scientists involved to the conclusion that the remains from this grave belonged to the Romanovs, minus 2 Romanov children, and the maid, cook, valet, and doctor who died with them. Mystery solved, identities restored, right? No, not quite. Not everyone was convinced. The 2 missing Romanov children kept a question alive. Couldn't any of them have escaped? In part 2, we're going to talk about one of the most famous royal mysteries of all time. The DNA drama is just getting started, so thank you for watching, and I hope to see you again for part 2 of the deepest dive into Romanov DNA that no one asked for. We have skeleton 3. I misspelled skeleton. Welcome to part 2 of the deepest dive into the Romanov DNA story that nobody asked for. In part 1, we talked about a grave discovered near Yekatarinberg in Russia, where the lives of Tsar Nicholas II, his wife Tsarina Alexandra, their 5 children, and the family's 4 servants met a violent end in July of 1918. Archaeologists excavated the remains of 9 adult individuals from this grave, and DNA analysis identified Nicholas, Alexandra, and 3 of their daughters. Not everyone was convinced by these results and the conclusions drawn from them, and various critiques came out after the publication of that first Romanov DNA paper. The grave only contained 9 individuals instead of the expected 11. If these are the Romanovs, 2 of their children are missing. This discrepancy was interpreted in multiple ways. Some people argue that these couldn't be the Romanovs at all, because the number of people recovered from the grave doesn't match the number of people who we know were executed with Tsar Nicholas in Yekaterinburg. However, this argument is based on the assumption that everyone had been buried together in the same grave. It's also based on the assumption that when the Romanovs and their servants were herded into the basement of the Apadiev house, none of them left alive, and none of them escaped. After the execution in 1918, the Bolsheviks only reported the death of Tsar Nicholas. They claimed Tsarina Alexandra and the couple's only son, Tsarevich Alexei, had been sent away from Yekaterinburg. But they didn't mention Nicholas and Alexandra's 4 daughters, the Grand Duchesses Olga, Tatiana, Maria, and Anastasia. They also didn't produce any corpses. With no bodies to prove that Nicholas, Alexandra, and their children had died, rumors spread that some of them had escaped. This was nothing new for a case in which members of a royal family have disappeared under mysterious circumstances, and it's a theme we've explored on this channel before with the stories of Prince Louis Charles of France and Caspar Hauser. When rumors like this get started and people appear claiming to be a missing member of a royal family, there can be some really serious consequences. This isn't even the first time something like this has happened with Russian royalty. At least 3 different people claim to be Dmitry Ivanovich, son of Ivan the Terrible, and they're referred to as false Dmitries. There's also Emelian Pugachev, who claimed to be Tsar Peter III and led a rebellion against Catherine the Great, who had usurped the throne from Peter. If you don't know that whole story, absolutely look into it, it's wild. People did not wait long to begin claiming they were Romanovs who had escaped from Yekaterinburg. As early as autumn of 1918, only months after the execution, a woman appeared claiming to be Anastasia. Other people popped up saying they were Tsarina Alexandra or Tsarevich Alexei, but the most famous claimant arguably the world's most famous royal claimant ever, spent over 30 years fighting to prove her identity as a Romanov. The legal battle over this woman's identity led to the longest trial in German history. How did we get there? In 1920, a young woman was pulled out of a canal in Berlin. She was taken to an asylum and diagnosed with depression, but she didn't provide her name. While staying in the asylum, she saw a German magazine with an article about the Russian Grand Duchesses Olga, Tatiana, Maria, and Anastasia. And in 1921, the woman revealed her identity. She was Grand Duchess Anastasia Romanov. Or maybe Grand Duchess Tatiana. At first, there was some confusion over which Grand Duchess she said she was, but eventually she started going by the name Anastasia Tchaikovsky. This name came from part of her story about how she escaped from Yekatarinberg. She wouldn't use the name Anna Anderson until she was checking into a hotel in the United States and used a false name so she wouldn't attract attention. Despite that hotel episode, the claims made by the woman calling herself Anastasia Tchaikovsky attracted a huge amount of attention. This included a lot of news and press coverage. She convinced a lot of people that she was Grand Duchess Anastasia, that she had escaped Yekatarinberg. How did she do that? She knew so much detail about the Romanovs, details of the lives that people thought she could only know if she really was a Romanov. Some people said she had the same eyes as Anastasia, the same eyes as her father Nicholas. She also shared a specific physical characteristic with Anastasia called Hallux valgus. This is the medical term for bunions. I told you bunions would get involved. If you thought I won't use any opportunity I can find to teach you something about the human body, you were wrong. Let's talk about feet bones. Hallux valgus happens when extra pressure on your big toe pushes it out of alignment. The big toe ends up being pushed towards your other toes, and the misalignment really shows up at the metatarsophalangeal, or MTP, joint. The human foot is made up of an absurd number of bones, and they're divided into 3 groups. You have your tarsals, metatarsals, and phalanges, your toe bones. The singular form of phalanges is phalanx, like the ancient Greek battle formation. The metatarsophalangeal joint is where metatarsal 1 meets what's called the proximal phalanx of the big toe, and this is where bunions form with Hallux valgus. So we divide the foot into 3 groups of bones. You have your tarsals down in here, your metatarsals, these long skinny ones, and then your phalanges. So when we're talking about Hallux valgus and bunions and the metatarsophalangeal joint, we're talking about this joint right here. So you have your first metatarsal and your proximal phalanx of your big toe. And this foot happens to actually be doing what Hallux valgus does, where your big toe is being pushed in towards the rest of your toes. So very glad that this cast just happened to do what I was trying to demonstrate. So yeah, there's feet bones for you. And bunions. So, is there something about Hallux valgus and bunions that makes this a useful characteristic for comparing Anastasia with someone claiming to be her? Is this a rare condition? No, not really. Anyone can develop bunions because they have various different causes. People can be born with bunions, which is referred to as congenital Hallux valgus. Wearing shoes that crowd the toes close together the way a person walks, or the inflammation caused by rheumatoid arthritis can create that extra pressure that results in bunions. You're also at a higher risk for developing bunions if you have a history of foot injuries or your biological parents have bunions, which prompts the question of genetics playing a role in Hallux valgus. This is not the video for that discussion, but all of these different causes mean Hallux valgus and bunions are common. It has been estimated that up to 1/3 of Americans have bunions, so it's not a particularly useful trait in this situation. Not everyone was convinced by the bunions or what this woman knew about the Romanovs and their lives. She had access to a lot of information that had been published about Russia's imperial family, so people argued that she didn't have to be Anastasia or any of the other Grand Duchesses to have known all of these details. Enough people discussed her claims that they wrote and published books about her during her lifetime, and the first Anastasia survival film came out in 1928. The woman calling herself Anastasia spent several years moving around between different hospitals and living with people who believed her and supported her, often people who had left Russia as a result of the revolution. For a while she lived with Duke Georg Nikolaevich de Beauharnais of Leuchtenberg. He was a great-grandson of Tsar Nicholas I of Russia, and a descendant of Eugene de Beauharnais, the stepson of Napoleon Bonaparte. I really cannot. He's just everywhere. Duke Georg and his ancestor Eugene are also distant relatives of Grand Duchess Stephanie de Beauharnais of Baden, who we met in another story of a royal claimant, Caspar Hauser. What's the point of claiming to be a lost member of a royal family? As we've seen, these claims could attract a lot of attention. Some claimants started rebellions in attempts to put themselves on the thrones they declared rightfully belonged to them. Supporters might give a claimant money or gifts or provide them with somewhere to live. In the case of the Romanovs, there was the potential to gain access to a lot of money. This money had been deposited in a bank in Berlin by Tsar Nicholas II. In her efforts to claim this money, the woman calling herself Anastasia Tchaikovsky got involved in a legal battle that lasted 37 years, the longest in Germany's history. Her appeal was rejected because the court decided she hadn't provided enough evidence to prove that she was Nicholas's daughter, Grand Duchess Anastasia, but a verdict wasn't released until 1970, the same year the album Black Sabbath was released. By that time, she no longer lived in Germany. In 1968, she moved to the United States and married a man named Jack Manahan, changing her name to Anastasia Manahan. Anastasia Manahan died on February 12, 1984 in Charlottesville, Virginia, and her remains were cremated. A decade would pass before DNA could put her claims to the test. So now we've caught up with the publication of the first Romanov DNA study in 1994. Now we have genetic data from the Romanovs, though there's still more controversy about those conclusions. The availability of that data meant it could be compared to Anastasia Manahan's DNA. But her remains were cremated, which can destroy DNA. Where could anyone locate her biological material for DNA samples? Hospital archives and a bookstore in Chapel Hill, North Carolina. In 1974, Anastasia Manahan underwent surgery to remove a bowel obstruction. This surgery involved removing part of her intestines, and those parts were fixed in formaldehyde and preserved in paraffin wax in order to be kept by the hospital's archives as pathology specimens. Another legal battle erupted, this time over who got custody of these tissue samples for testing, but other samples were located too. Jack Manahan had preserved locks of his wife's hair in envelopes that ended up with his library, which was then purchased by a bookstore in Chapel Hill after his death. A woman named Susan Grindstaff Burkhart bought the envelopes containing the locks of hair for $20 and probably then took them home to show her husband, who happened to work in a DNA research lab. This is the craziest part of the story for me. A genealogist then put Susan in touch with Anastasia Manahan's biographer, Peter Kurth, and he made the arrangements for the DNA testing. 3 independent labs tackled DNA extraction and analysis of the preserved intestinal tissue and 6 hairs. The Forensic Science Service in the UK, Pennsylvania State University, and the Armed Forces Institute of Pathology in Maryland. Importantly, the hair still had roots. The follicle in the root of a strand of hair contains nuclear DNA. Again, that's the DNA that lives in the nucleus of your cells, and you inherit your nuclear DNA from both of your biological parents. The hair shaft contains mitochondrial DNA, which, as we've discussed before, lives in your cell's mitochondria and you inherit from your biological mother. The labs tested both types of DNA, and all 3 labs received the same results. First, they were able to determine that the hair and the preserved intestinal tissues came from the same person. That's a really good start, but what about a comparison with the individuals from the grave near Yekatarinberg who had been identified as the Romanovs? Let's revisit short tandem repeats real quick. These are short, repeated chunks of DNA that vary in number between individuals. These are inherited from our biological parents, so Anastasia Romanov would have short tandem repeats that match Tsar Nicholas and Tsarina Alexandra. The short tandem repeat, or STR, profile of Anastasia Manahan differed from the profiles of Nicholas and Alexandra in 4 places, excluding the possibility of Nicholas and Alexandra being her biological parents. 4 might not sound like a lot, but it's too many differences to establish a parent-child relationship. Similar results came from the mitochondrial DNA analysis. Anastasia Manahan's mitochondrial DNA sequence differed in 6 places when compared to the sequences obtained from Alexandra, the 3 daughters buried with her, and Prince Philip, Duke of Edinburgh, a female line descendant of Alexandra's sister Victoria. Again, this was too many differences in their DNA sequences. Anastasia Manahan, the world's most famous royal claimant, was not Grand Duchess Anastasia Romanov. If she wasn't Anastasia Romanov, then who really was this woman? During the 1920s, rumors of an alternative identity began spreading. Some people recognized this woman as Franziska Szankowska, a Polish factory worker who had gone missing in Berlin in 1920, right before the woman who would become Anna Anderson or Anastasia Manahan started claiming to be a Russian Grand Duchess. To explore the question of whether or not Anastasia Manahan was really Franziska Szankowska, Franziska's great-nephew Karl Maucher donated a sample to the Forensic Science Service laboratory for DNA testing. Karl was the son of Franziska's niece and the grandson of her sister, so he would have the same mitochondrial DNA sequence as Franziska. Okay, so here's what our mitochondrial DNA family tree for Franziska Szankowska is going to look like. We'll put Franziska here. And she had a sister, Gertrude. Gertrude had a daughter, Margareta. And then Margareta had a son, Karl. Karl's mitochondrial DNA sequence didn't match the sequence of Tsarina Alexandra and her family. Good, that's what we would expect if they're not closely related. There were 5 identical matches between Karl Maucher and Anastasia Manahan, suggesting that he could be her maternal relative. When compared to mitochondrial DNA sequences in databases, the sequences shared by Karl Maucher and Anastasia Manahan were rare. They didn't match any of the sequences in the database. This rarity further supported the idea that these two individuals were related to each other. Statistical analysis put the probability of the match between Karl Maucher and Anastasia Manahan being completely random as less than 1 in 300, and that there was a 98.5% probability that Anastasia Manahan and Franziska Szankowska were the same person. Additionally, Karl's mother, Margareta Ehrlich, donated a DNA sample to the University of California at Berkeley for testing. She was the daughter of Franziska's sister Gertrude, and her sequence matched both Karl Maucher and Anastasia Manahan. Ultimately, these results support the hypothesis that Anastasia Manahan or Anna Anderson and Franziska Szankowska were the same person. So that solved one Romanov mystery. However, there was still more controversy surrounding that grave near Yekatarinberg. Did those remains truly belong to the Romanovs? If they did, where are the 2 missing Romanov children? In part 3, we'll meet members of the extended Romanov family as teams of geneticists challenge the original Romanov DNA paper. I hope you'll join me for the resulting drama. Thank you for watching. The fate of the Romanovs had different teams of scientists beefing it out for half a decade, claiming the other teams were wrong and conducting their own DNA tests. The results and conclusions from the first 2 Romanov DNA papers, the identification of the remains from the grave near Yekatarinberg, and the comparison with the DNA of Franziska Szankowska, who had claimed to be Grand Duchess Anastasia, were not enough to convince everyone. 2 Romanov children were still missing, but the second half of the 1990s, when the field of ancient DNA studies was still very new, saw a flurry of more Romanov DNA papers and one mysteriously unpublished paper. These tests focused on Tsar Nicholas and his wife, Tsarina Alexandra, and they sought to answer the same question. Had the last emperor and empress of Russia really been found? The skeletal remains of 4 adult men were excavated from the grave in the forest near Yekatarinberg. 3 male servants have been executed alongside Tsar Nicholas and his son, 13-year-old Tsarevich Alexei, the physician Dr. Eugene Botkin, the valet Alexei Trupp, and the cook Ivan Kharitonov. The identification of one of these skeletons, labeled as skeleton 4, as Tsar Nicholas, rests on 2 types of DNA. Short tandem repeats found in nuclear DNA, DNA in your cell's nuclei that you inherit from both of your biological parents, and mitochondrial DNA, the DNA in your cell's mitochondria that you inherit only from your biological mother. Skeleton 4's short tandem repeats matched 3 of the grave's female skeletons in patterns that we would expect if skeleton 4 was their biological father. This fits with the Romanovs if those 3 skeletons belong to 3 of Nicholas's 4 daughters. Skeleton 4's mitochondrial DNA sequence matched 2 living maternal relatives of Tsar Nicholas, except for one thing: a mutation called point heteroplasmy. Heteroplasmy is when an individual has 2 or more distinct mitochondrial DNA sequences in their cells. Point heteroplasmy is when this happens at a specific point, a base, also called a nucleotide, in the sequence. When we talk about a DNA sequence, what we mean is the bases, the nucleotides adenine, guanine, thymine, and cytosine, symbolized by the letters A, G, T, and C, lined up together in a specific order. So at each position in the sequence, you'll find one of these 4 nucleotides, but not in the mitochondrial DNA sequence of skeleton 4, identified as Tsar Nicholas. At position 16169, Nicholas had a mixture of the 2 bases cytosine and thymine, C and T. The 2 living maternal relatives to whom his DNA was compared did not have this. At this time, heteroplasmy was thought to be really rare. In order to verify that Nicholas had this point heteroplasmy, and that skeleton 4 really is Nicholas, we need to look at DNA from someone more closely related to him. Grand Duke George Alexandrovich Romanov didn't live to see the Russian Revolution and his family's fall from power. Born in 1871, he was Nicholas's younger brother, and when Nicholas became emperor, George was actually the heir to the throne because Nicholas hadn't had children yet. However, George was diagnosed with tuberculosis, and there wasn't a treatment for this disease at the time. Honestly, it can still be hard to treat tuberculosis now. At his doctor's suggestion, George went to live in the spa town of Abbas-Tuman, which is now Abbas-Tumani in Georgia, in the hopes that the dry mountain climate would help improve his health. It didn't, and he died there in 1899. George was buried in the Saints Peter and Paul Cathedral in St. Petersburg. This means we have someone closely related to Nicholas, who would share his mitochondrial DNA sequence with a known burial location. Scientists took samples from George's femur and tibia, and the article published about this actually has photos of the bones after the samples were taken. The bone samples they took for DNA extractions are huge, and I wanted to bring this up as a way of just briefly reflecting on how far the field of ancient DNA has come over the past 30 years. When we drill bones for DNA samples now, we don't take that much, and we tend to drill from parts of the skeleton that won't be as noticeable as a giant chunk missing from a femur. We're often drilling teeth, though in the case of George and Nicholas, samples had been taken from the femur and tibia of skeleton 4, Nicholas, so this gave the scientists a consistent sampling strategy. These tests included another comparison with the mitochondrial DNA sequences of 2 living relatives of Nicholas and George. These were Countess Xenia, a descendant of their sister, who was also named Xenia, and James George Alexander Bannerman Carnegie, the 3rd Duke of Fife and Chief of Clan Carnegie. He was a descendant of Nicholas's aunt, Queen Alexandra of Great Britain, who was the sister of their mother, Tsarina Maria Feodorovna. Now that we're starting to get some more characters in this part of the story, let's start adding them to our family tree here. So here's George. Nicholas and George had a couple sisters, one of whom was Xenia. And if we go down her family tree of female descendants, we then get Countess Xenia. For our other living maternal relative at the time of this study, we need to go back up to Nicholas and George's grandmother, Louise of Hesse-Kassel, Queen of Denmark. So one of her other daughters was another Alexandra, the daughter-in-law of Queen Victoria and thus later Queen of Britain herself. So through her female line descendants, we get the Duke of Fife. Okay, so now this is our completed mitochondrial DNA family tree for Tsar Nicholas. Their mitochondrial DNA sequence matched the sequences from the individual identified as Tsar Nicholas, except for the point heteroplasmy. Neither Countess Xenia nor the Duke of Fife had this mutation. So what about George? George's mitochondrial DNA sequence matched the sequences of Countess Xenia and the Duke of Fife, except for the point heteroplasmy. At position 16169, George had the same mixture of the nucleotides cytosine and thymine observed in skeleton 4, the individual identified as Tsar Nicholas. Both brothers having the same mutation in their mitochondrial DNA at the same position in their sequences makes it likely that the mutation occurred in their mother, Maria Feodorovna, and then she passed it to them. But we can't really know that with more certainty without a DNA sample from her too. Further studies also showed that mitochondrial DNA heteroplasmy is more common than previously thought. You would think that these results would make for a much more convincing positive identification of the remains of Tsar Nicholas, but no, apparently not. More controversy came from samples of biological material collected from Nicholas before his death. In 1891, before Nicholas became Emperor of Russia, He was visiting Japan when he was attacked by a random dude wielding a sword. The bloodstained handkerchief that had been used to treat the resulting head wound ended up at a Japanese museum. In 1997, a team of scientists claimed to have successfully extracted and analyzed DNA from the bloodstained handkerchief that didn't match the DNA profile previously obtained for Nicholas. But they didn't publish their study or go through any peer review process. We've seen this before with some of the DNA studies of Kaspar Hauser that also use bloodstained fabric as their source of DNA. Without a published article, we can't know what extraction protocols they used or what kind of decontamination procedures their lab had in place. We can't know their methods, what results they obtained, or how they reached their conclusions. The peer review and publication process is how dialogue takes place within scientific communities. It's part of how we learn what methods work and what doesn't work, and why. This has a huge role in how we design new projects. It really is a big part of how we communicate with each other, and it's not unusual to be asked, hey, do you have an article or citation for different methods and workflows for analyses or tools? That's why it's super weird and frustrating to learn that a study was done in response to an initial study, that the results challenged the results and conclusions of that initial study, but it wasn't published. Things get even weirder. That same lead author of the not published bloodstained handkerchief study then led another project trying to extract mitochondrial DNA from 25 hairs attributed to George. They claimed the sequences obtained from these hairs didn't match the mitochondrial DNA sequences obtained from Nicholas, and that it lacked the point heteroplasmy observed in the DNA that came out of George's bones. These results could be the result of contamination. They might have been looking at the DNA of someone who wasn't George, but it's difficult to know. They did publish this paper, but they published it in a Japanese journal, so it's difficult to access and there doesn't appear to be an English translation. I wasn't able to get access to the original study, so the reference I have for it is in a different paper that cites it. Another team of scientists then launched another study and published a paper that starts with claiming that the samples used in the original Romanov DNA study were contaminated. Their big problem with the original study was the size of DNA fragments obtained from the 9 individuals buried in the grave near Yekatarinberg, so it's time to beef it out over base pairs. We've already talked about the 4 nucleotides that create DNA: adenine, thymine, guanine, and cytosine, which we also refer to as bases. These create the 2-stranded double helix structure we all know and love by pairing up with each other. Adenine pairs with thymine, and guanine pairs up with cytosine. When we talk about the length of a DNA sequence or the size of a gene, we measure it in terms of how many base pairs long it is. One of the things that makes ancient DNA so difficult to work with is the fact that DNA degrades over time. The sequence falls apart, the bonds that hold nucleotides together break, and we're left with fragments, which again, we measure in the number of base pairs. With ancient DNA, we can expect fragments that are maybe a few hundred base pairs long, but we need to remember that factors such as the environment or how samples are handled and stored can influence how well ancient DNA is preserved. I'm saying that now because we're going to come back to it later. In the original Romanov DNA paper, they obtained DNA fragments up to 1,223 base pairs long. This new team of scientists, Knight et al., claimed that those fragments were way too big to be ancient DNA and challenged those results and conclusions with their own study. For the subject of their study, Knight et al. Chose Tsarina Alexandra's older sister, Grand Duchess Elizabeth Fyodorovna. Born in 1864 and nicknamed Ella, she married Grand Duke Sergei Alexandrovich Romanov, the brother of Tsar Alexander III and uncle of Tsar Nicholas II. After Sergei's assassination in 1905, Ella founded a convent, the Order of the Saints Martha and Mary. She was arrested by the Bolsheviks in April 1918 and was executed with other members of the extended Romanov family in July, shortly after the execution in Yekaterinburg. Ella and her companions had been thrown into a mine shaft during their execution, and their bodies remained there until they were exhumed in October. The bodies hadn't decomposed too much, so they were still recognizable enough to be identified, and they were autopsied. Ella was initially buried in a cathedral near the execution site, but her remains were moved to the St. Seraphim of Sarov Church in Peking in 1920, and then moved again to the Russian Orthodox Church of St. Mary Magdalene on the Mount of Olives in Jerusalem. She was canonized by the Russian Orthodox Church outside Russia in 1981, and canonized again by the Moscow Patriarchate in 1992. There's a figure of Ella carved over the west door of Westminster Abbey. In 1981, Ella's coffin was opened, and a bishop of the Russian Orthodox Church abroad removed one of Ella's fingers to keep as a relic in a reliquary. This relic was then taken to New York, and that's where samples were obtained for DNA testing. The DNA samples were extracted and analyzed at 2 independent labs, a lab at Stanford University and Los Alamos National Laboratory. Yes, this is the same place where the Manhattan Project developed the atomic bomb. They weren't able to obtain DNA fragments longer than 250 base pairs. The sequences of mitochondrial DNA they obtained from Ella didn't match Alexander's sequence. Remember, they're supposed to have the same mitochondrial DNA sequence they inherited from their mother, Princess Alice of Great Britain. Thus, they argue that the Romanovs may have been misidentified. So, what's going on here? How can we make sense of this? Thankfully, an article published later critiqued this study, as well as the 2 studies challenging the identification of Tsar Nicholas, the studies with the blood-stained handkerchief and the hairs attributed to his brother, Grand Duke George. So, what did this article have to say about all of this back and forth contamination and base pair drama? First, this article explained that a mixture of mitochondrial DNA sequences had been found in the Ella study. That sounds like a contamination red flag, and the sequences they obtained also didn't match Prince Philip, Duke of Edinburgh, the maternal relative to whom Alexander's DNA was compared. If we think back to the post-mortem treatment of Ella's remains and compare it to this grave excavated near Yekatarinberg, there were more opportunities for contamination. First, Ella's body was removed from the mineshaft where she had died, identified, and autopsied before being placed in a coffin. Then her remains were moved multiple times to different environments, and these different environments will have had a role in the preservation of Ella's DNA. Different environmental and storage conditions, including climate, temperature, and soil conditions, can preserve DNA differently, influencing how fast it degrades over time. Cold environments preserve DNA better, and it's similar to how certain environments can preserve soft tissue and create natural mummies. The grave near Yekatarinberg isn't in Siberia, but it's right next to Siberia, and the individuals stayed in that grave for decades. Their DNA might not have degraded as fast as Ella's DNA, resulting in those longer DNA fragments. Ella's remains encountered additional opportunities for contamination when her coffin was opened for the removal of some of her remains as relics, which were then again moved to a different location. There are also some odd characteristics in the overall research design of the studies of DNA from Ella's finger, Nicholas's bloodstained handkerchief, and George's hair. They didn't evaluate their data using the same framework as the original Romanov DNA study or the study of George's bones. The original studies tested 2 opposing hypotheses. The remains from Yekatarinberg are the Romanovs, or they are not the Romanovs. Part of this involved a lot of statistics, but they also included verified positive control samples from living relatives of the Romanovs. The finger, handkerchief, and hair studies didn't do this. They didn't introduce new samples from other living relatives, and if the authors of the Ella study are arguing that the samples from Yekatarinberg are contaminated, then that implies they must be contaminated by an individual who just happened to share the same maternal lineage as Prince Philip, who has the same maternal lineage as Tsarina Alexandra. Yeah, it's a bit of a stretch. The finger, handkerchief, and hair studies didn't really overturn the results of the original studies. However, there is still lingering controversy. 2 Romanov children, Tsesarevich Alexei and one of his sisters, are still missing at this point in the story. The discovery of a second grave near Yekatarinberg and the DNA analysis of its occupants will be discussed in part 4 of this series. But those studies are not the end of the Romanov DNA story. A couple of very recent studies will bring everything full circle, so I hope you'll join me for the next chapter. Thank you for watching. In 2007, a grave was discovered near Yekatarinberg, Russia. It contained 44 bone fragments and teeth that had showed evidence of having been damaged by fire and possibly sulfuric acid. This grave sat 70 meters from the grave that contained 9 individuals, identified as the last Tsar of Russia, his wife, 3 of their 5 children, and the 4 servants who had died with them in 1918. When the first grave was excavated, some used the fact that 2 of Nicholas and Alexander's children weren't found in the same grave to argue that these could not be the remains of the Romanovs. As we've discussed before, that argument is based on the assumption that they all shared the same grave following the execution. There was also a lot of disagreement over who was missing. It was clear that Tsesarevich Alexei hadn't been buried in this first grave. all of the skeletons in the first grave belonged to adults. Their bones had completed fusion and development. We would refer to them as having reached skeletal maturity. Alexei was only 13 when he was executed, so his bones would look very different from those of an adult. Skeletal analysis of the remains found in the second grave and testing the amelogenin gene showed that it contained 2 individuals, one of whom was a male between the ages of 10 and 14. It's the identity of the missing daughter of Nicholas and Alexandra that caused the disagreements. The couple had 4 daughters, the Grand Duchesses Olga, Tatiana, Maria, and Anastasia. The forensic anthropologists from the United States who examined the remains from the first grave thought Anastasia was missing, but the Russians thought Maria was missing. The other individual in the second grave was a female between the ages of 18 and 23. Maria was 19 and Anastasia was 17 at the times of their deaths, but what about a genetic relationship between the occupants of that second grave? The scientists carried out the same types of DNA tests for the second grave's occupants that were done for the first grave: short tandem repeats, or STRs, and mitochondrial DNA. These tests were carried out by 2 independent labs, the Armed Forces DNA Identification Laboratory in Maryland and the Institute for Legal Medicine in Innsbruck, Austria. First, they compared the short tandem repeat patterns of the 2 individuals from the second grave to each other. We share half of our STRs with our biological mother and the other half with our biological father, so we should share some of those chunks of DNA with our siblings, right? These 2 individuals shared a high degree of STRs and alleles, which are different variants of genes, suggesting a close genetic relationship. Their mitochondrial DNA sequences also matched each other, which is what we would expect from siblings who share the same biological mother. This study also involved retesting the individuals from that first grave identified as Nicholas, Alexandra, and 3 of their daughters, and again it used descendants of the maternal lineages of Queen Victoria, Alexandra's grandmother, and Empress Maria Feodorovna, Nicholas's mother, as references for the mitochondrial DNA sequences. When compared to living maternal relatives of Maria Feodorovna, the sequence from the skeleton identified as Nicholas matched them again, except for the heteroplasmic mutation at position 16169. They named this sequence the Dagmar mitotype. Nicholas's mother was born Princess Dagmar of Denmark, and it was usual for women marrying into the Romanov family, especially to the Tsar or the heir to the Russian throne, to change their names. They also didn't find anyone with a matching mitotype in a Danish database for mitochondrial DNA, indicating that this sequence is rare. The mitochondrial DNA sequences of the individuals identified as Alexandra and 3 of her daughters matched each other, replicating the results of the original study, and they matched both individuals from the second grave. The 2 individuals from the second grave also had mitochondrial DNA sequences that matched living relatives from Queen Victoria's maternal lineage, which was named the Queen Victoria mitotype. When they compared the Queen Victoria mitotype to databases that included mitochondrial DNA data from over 70,000 individuals, including populations from Russia and East and West Europe, they only found 1 individual who shared the Queen Victoria mitotype. This rarity supports the idea that Alexandra, her 3 daughters, and the 2 individuals from the second grave are maternally related to each other. When they retested the short tandem repeat profiles of the individuals from the first grave and compared them with the occupants of the second grave, they obtained a distinct profile for each person. So, we have 7 distinct individuals, and each of the 3 daughters and the 2 individuals from the second grave shared half of their genetic variants with Nicholas and the other half with Alexandra. This is consistent with parent-child relationships and the biological kinship relationships of immediate family. But we're not done. There's more DNA ground to cover. They also tested the Y chromosome short tandem repeats of Nicholas and the male individual from the second grave. These are repeating chunks of DNA that live on the Y chromosome, so they're passed from father to son, and that makes them useful for tracing paternal lineages. Living relatives were found among the descendants of Tsar Nicholas I. He was the great-grandfather of Tsar Nicholas II, so his male descendants, including Tsarevich Alexei, would share the same Y haplotype. Okay, so what about our YSTR family tree for Tsar Nicholas? I'm going to use a different color here for this. We're getting fancy now. So Nicholas is going to share his Y chromosome STR patterns with his only son, Alexei. And he would share it with George as well. That just wasn't tested. So, we need to go up to Nicholas's father, Alexander III, his father, Alexander II, and then the first Nicholas. And then from that first Nicholas, we have male line descendants who donated their DNA samples for comparison with Nicholas and Alexei. Named among those male line descendants who we got a comparison sample was Andrew. The Y chromosome haplotype of Prince Andrew Andreyevich Romanov, a male line descendant of Tsar Nicholas I and a distant relative of Tsar Nicholas II, matched both the individual identified as Nicholas II and the young male individual from the second grave. You might be thinking that all of this is enough DNA evidence to establish the identity of Nicholas's remains, but wait, there's more. A blood-stained handkerchief wasn't the only artifact from the day Nicholas was attacked by a random sword-wielding dude in Japan. The shirt he had worn that day, now covered in bloodstains, had been preserved in the archives of the Hermitage Museum in St. Petersburg. The scientists took samples from 4 different locations on the fabric and extracted DNA at 2 labs, the Vavilov Institute of General Genetics in Moscow and the University of Massachusetts Medical School. They found multiple different mitochondrial DNA sequences, which does indicate contamination, but one of those sequences matched the skeleton identified as Tsar Nicholas, matched his maternal relatives, and had the cytosine and thymine point heteroplasmy at position 16169 found in both Nicholas and his brother George. The Y chromosome short tandem repeat profile from the shirt's blood samples also matched the skeleton. So far, this is a considerable amount of data validating the results of the original study and supporting the hypothesis that the second grave contained the remains of Tsarevich Alexei and one of his sisters. To further explore whether or not the young male individual in the second grave was Alexei, the scientists look for genetic evidence of the blood disorder the last heir to Russia's throne carried during his short life, hemophilia. After he was born in 1904, it became very clear that Alexei had hemophilia. When his umbilical cord was cut, his navel, his belly button, wouldn't stop bleeding. His blood wouldn't clot. When a person has hemophilia, they don't have enough proteins called clotting factors that allow their blood to clot as part of the healing process after an injury. A person with hemophilia might bruise easily, or a small cut might result in a lot of bleeding. There are 3 types of hemophilia, called A, B, and C, and each type is low in a different clotting factor. Hemophilia A and B are the most common types. Hemophilia can be acquired if a person's autoantibodies, which are proteins made by the immune system that attack healthy tissue, start attacking one of those clotting factors, clotting factor 8. However, hemophilia is a genetic disorder, so it can be inherited or be the result of a spontaneous gene mutation early in fetal development. At the time of Alexei's birth, it was recognized that it ran in families. Specifically, it ran in the family of Britain's Queen Victoria and had been passed to her descendants. It's often referred to as the royal disease, but it did have tragic consequences and no treatment existed at the time. So, how does inheriting hemophilia work? Okay, so we have these 3 types of hemophilia, A, B, and C, and each is low in a different clotting factor. Hemophilia A is low in factor 8, hemophilia B is low in factor 9, and hemophilia C is low in factor 11. Each of these factors gets their own gene that lives on the X chromosome, and genes come in different variants called alleles. So there's a variant of the factor 8, 9, and factor 11 genes that produces enough of their respective clotting factor, and a variant, or allele, of each of these genes that doesn't produce enough of that clotting factor. The X chromosome, where these genes live, is one of the chromosomes that plays a role in whether a person is biologically female, with 2 X chromosomes, or biologically male, with 1 X and 1 Y, although there are other combinations of these chromosomes. The first X comes from a person's biological mother. A biological father will contribute either a second X chromosome or a Y. The Y chromosome doesn't carry any genes for these clotting factors, so the combination of an X chromosome with one of those gene variants that causes hemophilia A, B, or C, and a Y chromosome that doesn't have genes involved in the production of any clotting factors will result in a male individual with hemophilia. There is a lot more that could be said about hemophilia, and it deserves its own video, but that is what is relevant for Alexei. So, how can we test human remains for hemophilia? The key is knowing which genes and which variants of those genes cause hemophilia. The scientists used multiplex target amplification with polymerase chain reaction, PCR, to target the F8 and F9 genes, the genes associated with the most common types of hemophilia, A and B. Polymerase chain reaction is a method for making copies of DNA, which makes it easier to study, but you can also use it to test for the presence or absence of genes in a sample. Combining that with massively parallel sequencing, which is the same thing as whole genome sequencing, and I don't know why we have multiple names for the same thing, the scientists looked at the sequences of those two genes in Alexandra, who would have passed these genes to her children, and the remains thought to belong to Alexei and his sisters. They found a mutation in the F9 gene of Alexandra, one of her daughters, the skeleton presumed to be Anastasia based on the skeletal age estimation, and the young male individual from the second grave. This mutation made the F9 gene less functional, resulting in hemophilia B, which for some reason is also called Christmas disease. I don't understand why, so I will need to go and look this up later. This supports the hypothesis that these remains belong to Tsesarevich Alexei, because we know he lived with severe hemophilia and shared that gene mutation with his mother and sister, whom we would refer to as carriers of hemophilia. All of this genetic data from the reevaluation of the remains from the first grave and comparison with the DNA tests of the two individuals in the second grave led the teams of scientists to this conclusion. All of the Romanovs had been found and identified. Tsesarevich Alexei and his sister, they identified her as Grand Duchess Maria based on the age estimation of the bones fitting with her known age, could be reunited with the rest of their family and their ancestors in the Cathedral of Saints Peter and Paul in St. Petersburg. This did not happen. This is not the end of the Romanov DNA saga, but in part 5 of this series, we'll see DNA studies that push the boundaries of molecular anthropology. Thank you for watching. On July 17th, 1998, the remains of the last emperor of Russia, Tsar Nicholas II, his wife, Tsarina Alexandra, and 3 of their daughters were buried in the Cathedral of Saints Peter and Paul in St. Petersburg. Since the time of Peter the Great, almost all of Imperial Russia's rulers have been buried there. In 2007, the remains of Nicholas and Alexandra's 2 missing children, Tsarevich Alexei and Grand Duchess Maria, were located and identified. But the plan to bury Alexei and Marita in St. Petersburg and reunite them with the rest of their family came to an abrupt halt in 2015. The Russian Orthodox Church asked for more testing, more proof of the authenticity of these remains. The Russian Orthodox Church canonized Nicholas, Alexandra, and their 5 children in 2000. They're considered martyrs. This means that their bones are relics, so the Church wants to be very sure of their authenticity, hence asking for more DNA tests. The investigative committee of the Russian Federation exhumed Tsar Nicholas, Tsarina Alexandra, and Nicholas's father, Tsar Alexander III, for the collection of DNA samples. They also took samples from the blood-stained clothes worn by Nicholas's grandfather, Tsar Alexander II, the day he was assassinated in 1881, and they tried to get access to samples from Alexandra's sister, Grand Duchess Elizabeth Fyodorovna. As we've discussed before, Elizabeth, also called Ella, is buried in Jerusalem, but it's not clear if the investigative committee actually obtained samples from her. In 2018, the investigative committee announced positive results for the comparison of the DNA of Nicholas, Alexander III, and living relatives from amongst the extended Romanov family. Their results supported the father-son relationship between Alexander and Nicholas. But they didn't publish a paper about it. They issued a press release about it, and a bunch of different news outlets reported on it, but there's no article that describes their methods. They could have used Y chromosome short tandem repeats, repetitive chunks of DNA passed from father to son on the Y chromosome, to establish that genetic relationship between the 2 men. That method was used to identify the remains of Nicholas's son Alexei, but it's also not clear if Alexander's DNA was compared with Alexei. The news reports don't mention it, and they also don't mention if extracting DNA from the bloodstained coat of Tsar Alexander II was successful. Both Alexanders, Nicholas, and Alexei would all share the same Y chromosome short tandem repeats, but there isn't much information available about these studies. These studies also didn't result in Tsarevich Alexei and Grand Duchess Maria being buried with the rest of their family. Their remains are still being held in state archives, but This is still not the end of the Romanov DNA studies. Having so much genetic data from the Romanovs available means that all of this data is still useful for answering questions about the past. For an article published in 2021, their DNA was used to verify the value of a relic, a watercolor portrait of Tsesarevich Alexei, with a lock of hair enclosed in its frame that's held by the State Historical Museum in Moscow. The researchers wanted to know whether or not the hair actually belonged to Alexei, so they tested 2 hair shafts from the portrait. Though the DNA in the hairs wasn't preserved very well, the mitochondrial DNA sequence obtained from them matched Alexei, Alexandra, and Alexei's sisters. This makes it entirely possible that those hairs belong to Alexei. Another recent Romanov study looks to improve upon the methods used for extracting and analyzing DNA from single rootless hair shafts. For this study, the scientists wanted to test a combination of whole genome sequencing and a DNA extraction protocol that's optimized for targeting ultra-short DNA molecules from single rootless hair shafts with the oldest, and thus very degraded, samples they could find. The oldest hair samples they could access came from 2 items held in a private collection: a pendant that had been made for Tsarina Alexandra, and a framed photo of Tsar Nicholas's maternal grandmother, Queen Louise of Denmark. They compared the mitochondrial DNA sequences obtained from the hairs preserved with these items to the published sequences of Alexandra and Nicholas. All of the sequences from the hairs in the pendant that had been made for Alexandra had the same mitochondrial DNA profile, and they belonged to the mitochondrial haplogroup H1AF2. A mitochondrial haplogroup is a collection of mitochondrial haplotypes, and those are defined by similar genetic variants in their sequences, and the people grouped together in a haplogroup share a common maternal ancestor. Alexandra belongs to haplogroup H1AF2, just like the hairs from the pendant. The hairs in the framed photo are presumed to have come from Queen Louise of Denmark, and the photo had belonged to her daughter, Tsarina Maria Feodorovna, and then to Maria's daughter, Grand Duchess Xenia, before being passed to other members of the family and then ending up in a private collection. Louise was Tsar Nicholas's maternal grandmother, so the mitochondrial DNA sequences from the hair in the framed photograph were compared to him. The sequences matched, sharing the same mitochondrial haplogroup, T2A1A, and sharing the point point heteroplasmy mutation at position 16169 in their sequences. So this means that if the hairs belonged to Queen Louise, then she carried the point heteroplasmy mutation. She passed it to her daughter, Tsarina Maria Feodorovna, again known before her marriage as Princess Dagmar of Denmark, and Maria then passed it to 2 of her sons, Tsar Nicholas and Grand Duke George. This mutation didn't last very long within this mitochondrial lineage, at least that's what it looks like with the data we have, and that's why we don't see it in the other descendants of Queen Louise who contributed DNA samples to previous studies. That's the last Romanov DNA study I have for you. For now. This story, from the excavation of that first grave up through the publication of this most recent article, has been going on for almost the entire lifetime of the field of ancient DNA studies. Why is any of this important? That first Romanov DNA study was, in the words of the paper's authors, the first major historical investigation in which both STR and mitochondrial DNA have been used as investigative tools. The use of DNA analysis in this extraordinary case provides a dramatic insight into the power of these techniques in solving historical questions. Nicholas, Alexandra, 3 of their daughters, their physician, and the 3 servants were the first historical individuals who had their identities restored to them through a combination of skeletal and DNA analysis. That first study set the stage not only for the identification of other historical individuals through DNA analysis, but for the use of DNA in forensics cases with degraded, poorly preserved DNA. The advancements in DNA extraction and sequencing methods and technology, and the application of those advances to the study of ancient DNA and skeletal remains from the archaeological record that made that first Romanov study possible, shaped the studies that came later, including the identification of Alexei and Maria and the reevaluation of those original studies. The most recent Romanov DNA study, with the hair from Alexandra's pendant and the photo of Queen Louise, pushed the boundaries of DNA technology and its application to degraded DNA. In that study, they managed to get nuclear DNA from those hair shafts. Usually, we're only going to get nuclear DNA from a hair's root, not a rootless shaft. The nuclear DNA results they obtained supported their mitochondrial DNA results, and I don't know if anyone has followed up on these methods and replicated their results with similarly degraded samples. But this can have significant implications for the field of forensic anthropology. Single-shed hairs are often encountered in forensic casework, and hairs collected in older forensic cases might end up being stored in conditions that are not great for the preservation of DNA. So if we can create better methods for getting DNA with degraded samples from single rootless hair shafts, that could really help with forensic cases. If you're interested in reading more about the Romanovs, I have several book recommendations. If you're curious about the whole dynasty, try the Romanovs, 1619 to 1918. For Nicholas and Alexandra's family, I really enjoyed the Romanov sisters, The Lost Lives of the Daughters of Nicholas and Alexandra. As part of my research for this series, I reread The Resurrection of the Romanovs, Anastasia, Anna Anderson, and the World's Greatest Royal Mystery, as well as Ella, Princess, Saint, Martyr, a biography of Grand Duchess Elizabeth Fyodorovna, Alexandra's sister. There's no shortage of books about this family. Those are just a few I've enjoyed. If you've made it to the end of this series, thank you so much for watching. Because we've made it so far in this series, I made a series because I needed to be a little bit extra about it. I thought, let's do something stupid. Let's see if I can fit the whole family tree that I constructed for this video series onto the board. Don't mind me, I'm just checking the cheat sheets that I bring with me when I film in here. I think there's not even space for me in here. I think this is everyone that we mentioned, everyone we talked about, at least within this family, these various dynasties. But actually, let's go back in, and we're going to highlight everyone whose DNA we probably looked at. We have DNA from Philip. We tried DNA from Ella. We have the whole family here, lost and found. Duke of Fife, George, Countess Senya, Andrew, and then possibly Maria Fyodorovna, and possibly Alexander III. I think that's everybody. Yep, so just going, no, not Maria Fyodorovna, possibly Louise. But yeah, there we go. I probably spent way more time putting all of this together than I ever needed to, but here we are. And again, if you've made it through all of this craziness, thank you for watching.